This is a production of Cornell University.
No problem. And thank you for the lovely introduction. And it's great to be here. And yeah, I've always been fascinated with how you turn genes on and off. And, my brief stint in industry, showed me what was going on with trying to genetically engineer plants for the field. And now I'm still working in the lab, mostly with Arabidopsis. But I'm hopeful that some of the tools that we're building here will have applications in the field in the future. And so, yeah, what are we doing? So I'm trying to build gene circuits, but why are we doing this? So as I'm sure I don't need to, tell most people probably in the audience, but we've got climate change to deal with. We've got a growing population and a finite amount of land. So we really need to increase crop productivity. But in many cases, we've got stagnation in crop yields. And some of these factors are, societal and economic, but some of them could be mitigated by improved strains of plants being produced. And so we would like to use new techniques, new advanced ways of doing genetic engineering to be able to achieve that. And so there are some grand challenges out there that could really boost productivity, but they're going to require a lot of work to achieve that and more tools for better genetic engineering. So some of these, hopes for the future would be things like nitrogen fixation in cereals. So only your legumes fix nitrogen through symbiosis with bacteria. Now, if we could get legumes fixing nitrogen, that could really decrease the inputs required into those crops and increase productivity. But the ability to do this in nature for legumes requires a lot of developmental biology, signaling, and cooperation with bacteria. And that's multiple different things that need to be altered to allow this nitrogen fixation trait. And so we need very sophisticated tools to engineer that. In the another case would be C4 rice. As I'm sure many of you know, there's C3 photosynthesis and then a more productive C4 photosynthesis, particularly at higher temperatures. And rice, one of the major cereal crops, does not use C4 photosynthesis. And to engineer that into rice would require both biochemical changes in metabolism, as well as developmental changes, and then also to separate the biochemical pathways into these different cell types in a way that does not occur naturally in rice, which is C3. And so again, we need really complicated spatiotemporal control over gene expression. Now, in development, in normal biology, there are many different factors that control where and when a gene is turned on and off. And you can just see from these diagrams here that, just the decision between being a non-hair cell and a root hair cell in the Arabidopsis epidermis of the root, that's a really, complex decision with many different factors, different transcription factors and other genes involved with that process, and a lot of regulation. In going over to humans and looking at some work from my time in Berkeley, just looking at the number of splicing factors and that interact with each other, this is a massive amount of different interactions for controlling gene expression that happen both at the transcriptional level and also at the post-transcription level. So, what hope do we have for really being able to engineer this ourselves in plants for custom traits? Well, at the moment, we're quite limited by what tools we have to genetically engineer plants. So, quite often people rely upon the 35S promoter, which is a strong constitutive promoter. But sometimes you don't want to express a gene at really high levels everywhere in the plant. So there are other options out there if you don't want to use 35S. You could use a slightly weaker, more dreary tired promoter, like the actin-2 promoter, that face a bit like me, based on the fact that it's gone midnight now. But again, that's constitutively expressed, so it's expressed across the whole plant. So you may want to turn to a stress-induced gene, such as from heat shock or infection, and these would allow for condition-specific responses. Or you may want to turn to a cell type specific promoter. So you turn it on in only one part of the plant based on a developmental cue, for example. Or maybe you'll want a chemically induced promoter, such as using the DEX, which we use often in the lab to artificially induce gene expression. So we have these different options, but what happens if you wanted to combine them? What happens if you wanted the high expression of 35S with the stress response of a virus-induced promoter? Perhaps you also want it only to be in one place, so for example, the root cortex. Well, it's very hard to engineer a promoter with all of these features. And it's also very hard to identify a promoter that naturally has all of these features, especially if you've got to do it in every single crop repeatedly, and perhaps the promoter that has these properties doesn't exist. And then if you decide, oh, this isn't what we want it to be a more moderately expressed promoter, and we want it to be the same cell location, but now we want it to be chemically inducible. Well, now you've got to start your search all over again. And so this is where synthetic biology comes in, and hopefully we synthetic biologists can make it so that we have both. We don't have to choose between these different properties, but we can get cell type and condition specific expression all combined into one using what we call gene circuits. So how can we hope to achieve this? Well, we're hoping to achieve this with something called Boolean logic. So Boolean logic is just a mathematical framework for thinking about true, false, binary data. And you can think of it in terms of this lovely Venn diagram here, where you have only green representing on, where you have both inputs turned on. So this could be demonstrated with a simple electrical circuit, such as here, where you need a complete circuit to turn the light on. And here we have two switches. Once both are switched on, the light comes on. We complete the circuit. And this is very easy to visualize for an electronic circuit, and we can also visualize it with a symbol here. This is the symbol for the AND gate, and here is a truth table where we can see the output is only on, represented by a 1, and both of the inputs are on. In gene expression terms, well, you could think of an AND gate being, maybe there's a gene where you need 2 different transcription factors to bind before the transcription activates. So we can try to apply these mathematical and electrical engineering concepts to gene expression. And there are many different logic gates that can exist. So in addition to the AND gate, where what we effectively see is a restriction of expression. If we're thinking about it in terms of gene expression, this is restricting where a gene is expressed based on the activity of 2 factors. Whereas the OR gate is a really nice example because we broaden the expression out. You could imagine that as 2 different transcription factors, and either could cause the expression of the output gene. And then we can also use repression. So a NOR gate is the opposite of an OR gate. So it is on unless you have one of the inputs present. So in this case, you might think of the inputs as being repressors, such as repressive transcription factors, and either one of them binding will turn the gene off. So in theory, we can use these design principles to try to design these things. But is this really relevant? Is it really, really realistic to do such a thing? Well, in nature, we already see expression pathways that look very much like Boolean logic gates. So if we go back to the undergraduate class example, the Nobel Prize winning lac operon for gene regulation, the understanding the gene regulation, that was very fundamental to our understanding of molecular biology. So in this pathway, what we do is we have lactose that effectively activates the pathway, and we have glucose that turns it off. But interestingly, glucose turns off the pathway even in the presence of lactose. Not perfectly, but to a large extent. And so you can imagine this circuit looking like this in terms of the Venn diagram. So you only have output with lactose, but with glucose is present, it overrides it. It's a dominant effect. It's a dominant negative effect. It overrides the activity of lactose. And this looks like an A NIMPLY B logic gate. So we can draw the symbol for that with lactose coming in, glucose overriding it. We can draw the truth table. So we only have lactose by itself activating the output. And even if you have both, it is off. This is a generalization. It's not perfect. So we can build simple, nature has built simple logical gates already. So it's not unfeasible that we can try to engineer the same ourselves to get more sophisticated control over gene expression. Now, the other thing is thinking about these things in a digital manner, on or off, really a realistic thing to do? Well, in plants, we do have binary systems. So the control of the floral developmental gene FLC is controlled through an epigenetic digital switch. So in this case, you have a wild type plant where it's expressing FLC and it does not flower, it flowers on a normal timescale. And then if you have the FLC mutant, it will flower early. And the natural regulation of FLC works where it progressively more and more genes, more and more individual loci epigenetically turn off FLC expression as you go through winter, and then that gets memorized during the spring. So you will have warm temperatures where you express FLC as a floral repressor, and this delays flowering. And then over time, the exposure of cold, you will build up the silence in it, each individual loci. So in a diploid cell, each individual gene copy is regulated independently of each other. And eventually, when you start to warm up, you then get that memory state locked in through the polycomb repressive complex mark. And so we know that digital switches can exist in nature. So when we were trying to design our gene circuits, we really wanted to look towards, can we replicate a binary digital system for building these complex gates? And so we turned to site-specific recombinases. So these recombinases recognize short sequences, about 35 base pairs long, and this is the FRT recombination target site sequence. And so this is sat in the genome, and all the sequence in red between these two sites could be of functional importance that we want to conditionally remove. So then when we express the recombinase, in this case FLP, we cut that bit of DNA out. So this leaves one FRT site in the genome, and then all the sequence in between them is now cut out and becomes circularized, and so is not functional in any way that is useful. And so we effectively remove it from the system. And now it's not a scalar system. We do get left with a single recombination target site. But this tool set of recombinases allows us to try and build up a complex set of gates. And so the first simplest type of gate is a simple yes gate, a one input switch. So we go from off to on as we express the recombinase. And so in this case, what we would do is we'd have a constitutive promoter driving a reporter gene, such as luciferase or GFP, and we would use a terminator in what would normally be the 5' UTR region to turn this reporter off. So the promoter might be a constitutive promoter, but it's no longer able to express this reporter gene. We flank this terminator with recombination target sites, and then only when we conditionally express the recombinase do we get that terminator removed and the reporter gene turned on. So nice on paper, very simple, but first we've got to build this system up. We've got to identify, well, what promoters are useful, what terminators will function in this way, which recombinases will actually work in our system. So, there's a lot of steps that need to be done to build this up. So we moved to a protoplast system. So this is a 96-wall plate-based protoplast system. I found it incredibly reproducible, which I know a lot of protoplast systems do have an annoying level of variability. So I highly recommend checking out the protocol that Adil Khan, a really talented researcher in the Lister lab, has shared online for this. It took a lot of optimization. And the way it works is we isolate protoplasts from the leaves. So these digest the cell walls away, we release the protoplasts, and then we use PEG to transform our plasmid DNA into them. We incubate them for 1 to 2.5 days, and then we perform a dual luciferase assay on them in a 96-wall plate format. And the plasmids we transform in have two different luciferase genes. So one encoding a renilla luciferase, and this is reporting our output levels of our gene circuit. So this is the activity level. And then we have a normalizer. This is firefly luciferase. Our normalizer, its function is to account for variability in protoplast survival in each well, transformation efficiency. So by normalizing the amount of vanilla over the amount of firefly, we're able to see a nice reproducible level of gene expression. So now comes to us actually building and testing the components we need for gene circuits. And the first component we need really is a nice terminator to block expression of vanilla luciferase. So as I just walk you through this first data slide, we have the actin-2 promoter driving vanilla luciferase, and this top construct here is our control construct. It's our constitutive vanilla luciferase, and this is our standard always-on plasmid. And so whenever you see a blue bar on a graph, that is at control. And so we use this repeatedly as a way of like checking for the standard level of expression we would expect. And then we built variants of this plasmid, which have the FIT recombination sites, but they also have different terminators. They have this synthetic transcription block. There's also the OCS terminator, a common terminator used in plant biotechnology. There is also the a fusion of the two terminators, as some studies have shown that can enhance terminator activity. We have the OCS variant, which is a truncation of OCS. We also have the cauliflower mosaic virus terminator, another common terminator used in plant biotechnology. And then we also use an upstream open reading frame, which has been shown to cause RNA decay in transcripts from previous work that I was involved with. So we tested a range of these in Protoplasts to see which one inhibited expression the best. And while the transcription block and the UOF did inhibit expression to some extent, it was not very impressive. But the OCS terminator and all its variants did. And so we've been able to turn our gene expression off with OCS quite effectively. To our surprise, the cauliflower mosaic virus terminator did not function very well. It did not inhibit gene expression. And at first this stumped us, but then my PhD student, Patrick, found this paper from over 30 years ago where they'd actually done a similar experiment, placing the cauliflower mosaic virus at different distances away from the promoter, and they found if it was close to the promoter, termination did not occur. And so it seems to be a location-dependent termination, which is very interesting from a mechanistic point of view, and also important for us to know as synthetic biologists that genetic components can be very location and context specific. And that's something I think we as synthetic biologists trying to build, engineer very complicated pathways like this, we need to keep that in mind when we're building, new plasmids and functions into plants. So this was great. We found a terminator that can turn the expression off. But for our work in protoplasts, what we would like to do is have a promoter, a constitutive promoter that we can include into certain plasmids to show the on state, to represent the on state. And so we tested a range of different promoters that could drive the recombinase, FLP, in the system and compared it to the expression of both the constitutive ARLUC plasmid as well as our OCS terminator. And so when we did that, we found that all the promoters we tested, NOS, TCTP, actin-2, and 35S, they all did create what we call the yes gate, the one input switch, and activate. But only the TCTP did it to levels comparable to our constitutive ARLUC. So that was really good. It's nice to see that that we've been able to build a one input switch. But we wanted to go back into stable plants, do this in whole organisms, and see how this behaves there. And so we built a one input yes gate there, and we use this time the heat shock inducible promoter to drive the expression of the recombinase. And for a reporter, this time we're using nuclear localized GFP. And as you can see, without a heat shock, there are no nuclear localized GFP in the roots. But when we do heat shock the sister plants, we see lots of GFP accumulating in the nuclei across the roots. So this was great. So we've got the basic components we need to build a one input switch and even work in vivo with this. So that's great. And then I got very excited. I got maybe a bit bold and thought, oh, well, we can finish this project off in a year. Spoiler alert, that didn't work. That was 2019 when I thought that. So immediately I decided to jump into building 2 input gates. I thought, well, we've got FLP working, we've got the basic commands working. I can just grab the most commonly used recombinase in all of biotechnology, CRE, part of the CRE lock system, and just use that as the second input with FLP. It's guaranteed to work, guaranteed to work. Sadly, it didn't work. And this was really surprising. So the way the OR gate was built, we put the recombination target sites for both FLP and CRE surrounding the OCS terminator. So either recombinase can cut the OCS terminator out. And then for each of the plasmids representing the 1 input state, we included either FLP or CRE. And we also included a 2 input state where we included both FLP and CRE. And when we look at this plot, you can see, again, our positive control in blue, our off state in gray, and then the expected on state in green. But only FLP behaves as expected. So again, we're seeing it activate as we would expect, but CRE 1 input does not. It's repressed still, which is really confusing. Even more confusing is that the inclusion of CRE on the plasmid also inhibits the activity of FLP. And so it's taking the plasmid that did previously have high recombination rates and causing no activity in terms of vanilla expression. So this was really surprising. And I think we're finally close to figuring out the problem here, but this was a big shock for us. So then we started looking for alternative recombinases to try and build up a 2 input switch. So we took that big step of testing different 1 input yes gates for another recombinase that could work. And so initially, my honors student from 2020, Florence, did an amazing job of testing lots of different recombinases, S-CRE, V-CRE, VICA, all of them are CRE relatives. However, none of them cause activation, not anywhere near as impressive as the activation seen by FLP. So again, this was really surprising to us that none of these behaved the way we expected them to. So I, in parallel with this, was testing and cloning B3, which is a relative of FLP. And when we tested it, we found that, yes, actually it behaves almost the same as FLP in the 1 input yes gate. So fantastic. Finally, we found a recombinase that can behave itself and work the way we would want it to, and start building the 2 input switches. But now I'm getting a bit of healthy paranoia. I'm thinking, well, maybe they're relatives of each other. Maybe they cross-react. In animal cells, they've been shown not to cross-react, but we've always got to take the literature, published literature, with a grain of salt. So I go ahead and test that. And I'm very happy to say there was no cross-reactivity. So when we put FLP on a plasmid with the B3 target sites, there's no activation, and same in reverse. So we get good activation of the appropriate target sites, but not with the inappropriate recombination target sites. So this is great. So now we can finally go back to building those 2 input gates that we initially tried to build. So we went back to the OR and now also expanding out to the AND gate. And so the AND gate, the OR gate follows the same design as with CRE, but now with the B3 recombinase and target sites. So again, either recombinase can cut it out. And the other difference to note here is that my paranoia was creeping in, and so I designed an extra 2 input version plasmid where I swapped the ordering of the input genes around, just in case that made a difference. It doesn't seem to make a difference, but again, I think it's important that we have as many different controls for the context of the components we're using in synthetic biology as possible. And so for the OR gate, we see that the 0 input state is repressed, and all of the 1 and 2 input states are expressed at least the same level as the constitutive or higher. So this is wonderful. We get OR gate logic, and this is brilliant. So then we tried AND gate, and the AND gate works where we have 2 copies of the OCS terminator, each one cut out by a different recombinase. So now we need both recombinases to be expressed in order to activate vanilla. And you can see here that when we have the 0 input state or the 1 input states, we have no expression or very low expression of vanilla luciferase. But if we express both of the recombinases, suddenly we get very high levels of renilla. After 24 hours, it's still lower than our control, but by 48 hours, it's at comparable levels, which is interesting. So it suggests that this reaction is slower than the OR gate, which might make sense because we need two different recombination events to occur on the same plasmid in order to get activation. So it might be the dynamics are different, but it still works. So that's really good news. So then we wanted to take it back into stable plants. We wanted to see if we could get an AND gate to work with natural or relevant inputs to switch between them. So we created another plasmid. This time we're using the 35S promoter. And again, we've got the two different terminators, each removed by different recombinase. We've got nuclear localized GFP as the output. We have a seed coat marker for selection of transformed plants. We have a constitutively expressed M cherry that is also nuclear localized. And this will mark every nuclei in the plant, and the root is where we're looking for expression. It will mark every root nuclei as red with M cherry. Then we used FLP as one of the inputs using the DEX inducible promoter. And then we also used a cortex specific promoter to induce B3. So we should only see expression of GFP in the cortex after DEX induction. And so you can see here with the solvent control, we have the bright field and GFP, we see no GFP nuclei. However, if we look where we add DEX, we do see many different nuclei lighting up green. And if we look at so I took a Z stack at the confocal of these multiple roots, and here's a representative where I also stained with PI to outline the cell walls. And so you can see on the outer layer, you have the epidermis, the cortex, you've got these bigger, rounder cells that represent the cortex, the second layer in from the root, and then you have the epidermis, and you can't really see the stele in this experiment. And you can see that the only nuclei in this and other examples that lights up green is in the cortex. So this is wonderful. So not only do we only see expression of green nuclei, observed green nuclei in the DEX treatment, but they're also only in the cortex. So this is a functioning AND gate working in vivo. So this gives us hope for being able to move this and build more circuits in stable plants, which is ultimately where we want to go. Working in Protoplasts is a nice, fast way of screening lots of things and working out what does work and what doesn't. But ultimately, we do want to be working with stable plants and potentially moving into crops. So now we've got activation working really, really well. Now, can we deal with repression? Can we turn off genes? And so I tried to build the NOR gate. And so this time to design a NOR gate, we don't use a terminator. In fact, we want it to be default on. So therefore, we want to turn it off. And in this case, we were removing the CDS of renylluciferase with either B3 or FLP. However, when we actually did the experiment, we saw that the no input state was on as expected. And FLP was able to turn it off quite rapidly. But after 24 hours with B3 alone, it doesn't change, it's not off. After 48 hours, we do get some decrease in expression. So B3 seems to be acting very slowly. It's not very effective, it's acting much slower than FLP. And at this point, I took it quite personally. I'm sure B3, being a molecule, has no motives to ruin my life, but I took it very personally because B3 had been the savior. It had rescued me from, Cree being a failure. And so I was very excited. I thought we'd be smooth sailing. Sadly, that was not true. So again, I relearned my lesson. We've got to test every component in isolation and look at different contexts where it may or may not work. So in this case, I isolated, I made NOT gates where we just have either FRT sites or B3 RT sites to be recombined out. And we can see that in the FLP, even after 24 hours, we get repression. But after 24 hours with the B3, there's no repression. mild repression after 48 hours, slightly better repression after 64 hours. So just as we saw in the NOR gate, when B3 is trying to cut out the Ranilla luciferase CDS, it's not behaving itself very well. It's very slow. It's very inefficient. And so I thought, well, what could possibly be going wrong? Is it the sequence itself? Is it something to do with the distance between the recombination sites? So in the terminator example, the recombination sites were 700 base pairs away from each other. In this case, they're 2 kb away from each other. So is there a way we could turn the gene off by removing a smaller region? And the actin-2 promoter used in these constructs is only 800 base pairs, so very similar in size to the terminator that B3 was very good at removing. So I redesigned the constructs to target the promoter region, and for both FLP and B3 circuits, we see good repression even after 24 hours. So again, a really great example of context-specific activity. So, you know, synthetic biology, we need to test parts, we need to explore things in detail. So now we've got this working, what I wanted to do next was to combine activation and repression into the same circuit. So this was going after the A NIMPLY B. So by going after the A NIMPLY B circuit, the way this would work is that the zero input state would be off because we still have the OCS terminator in the 5' UTR. If we activate FLP only, we can remove this OCS terminator, and that's exactly what we see. We see activation when we activate FLP. But then if we activate B3 or B3 and FLP, what we do is we remove the actin-2 promoter. So it doesn't matter if the terminator is removed, because if the promoter is removed, you're never going to get expression of RLOC. So, and that's exactly the behavior that we saw. So this was a really wonderful experiment and showed that we're able to build these more complicated circuits that integrate both activation and repression into it and replicate, you know, expression patterns similar to what we see in the lac operon and potentially in other places in nature. So we've been able to build these more complicated circuits, but there's still a lot to do. And one of the things that we looked at next was, can we, using just one recombinase, expand our toolkit for the circuits further? And so with FLP, it's already published a split site where you can split FLP into 2 inactive components and then dimerize to bring them back together. And so this is the crystal structure of FLP binding to DNA. And then what we did was we split it at a known split site, and here are AlphaFold 2 generated models of what these would look like. And so we have one fragment, you know, very large fragment 1 of FLP with a linker, the C1 homodimerization domain from phage, and a nuclear localization signal. And then we also have the other part fragment of FLP, a much shorter one, the linker, the nuclear localization signal, and the C1 homodimerization domain. So if expressed by themselves, neither one can cause activation, neither one will cause recombination. However, if you express them both in the same cell at the same time, they will reconstitute and form a new active recombinase and are able to activate. And so we put this to the test to try and build what we call a split AND gate. And so Florence, my amazing honors student, she was able to build and test this. And so you can see here, you just need one terminator site. So this reduces the size of the constructs. And you're able to remove this OCS terminator only when you have both parts of the recombinase expressed in the sample. So this is wonderful. And one of the advantages of this would be if you imagine you had one of your inputs where it's only expressed ever in the root, but the other one might be induced by heat shock. So that would be induced across the whole plant. But what happens if you wanted to get seeds from that plant and grow them in the next generation without the circuit already being triggered? With a traditional AND gate approach, it would already be partially triggered by the heat shock because I would recombine out one of the 2 terminators. In this case, you would never have the aerial tissue have recombination events because you need both recombinases be expressed at the same time in the same place. So for certain circuit applications where you won't have expression in the shoot-apical meristem, you would be able to save seeds from the next generation. So we think this could be a very useful modification to the system. Additionally, we also built a NAND gate, which was really great. And so Florence actually, she was bored after her honours and decided before she went to start her job to come back into the lab, which amazed me. I was still on Christmas break when she came back in, but she came in and she pushed this forward and designed a NAND gate. And so the NAND gate would cut out the promoter, but only when both halves are expressed. And this is exactly what we see. We see good levels of expression, except for when we express both halves of the recombinase. So now we can get AND and NAND gate logic using these recombinases. So I think this is a great example of how we can expand out the toolkits. And at the moment, we're doing research on how to do this further and create more split recombinases, as well as bring other recombinases like Cree back into our toolkit and be able to make much more complex circuits with more than 2 inputs. But yeah, so just before I wrap up the talk, I did want to talk a bit about the growing field of plant gene circuits in general. So we've had a few publications this year on plant gene circuits. So Jen Brophy at Stanford led this amazing study published in Science, where they created synthetic genetic circuits in plants using bacterial transcription factors. And so they were able to make a wide array of different logics by combining different bacterial transcription factors, both activators and repressors in plants. And so the advantage of this system and other systems like it would be, unlike my recombination-based system, these would be reversible. So the advantage of a recombination-based system is that once the recombination event happens, it is a stable, locked-in change. And so if you require that memory of the event, then recombination circuits are going to be very valuable. So for example, a dream scenario would be if, a farmer predicted like a particular disease or environmental stress was coming towards their field. If they had time, they could potentially spray their field. But with a reversible circuit based on transcription factors, the timing of that would be very key and might be very difficult to achieve. Whereas with a recombination-based circuit, you might be able to prime the plants ahead of the incoming stress. So that would be one of our hopes for how we could use this technology in the future. But my recombination-based circuits are not reversible. And so if reversibility is something important, then these sorts of circuits would be very important based on artificial transcription factors. And so another paper that's come out of the Lister lab, led by the really great Adil Khan, is a CRISPR interference-based gene circuit. And so that's going through peer review at the moment. But because I think it's such a really interesting system, I just want to take a moment to walk you through the basics of it. So the way this system works is that you built a NOR gate using a synthetic promoter. And so this promoter has had 2 artificial single guide RNA binding sites introduced to it. And if you express one or both of the single guide RNAs, then the CRISPR dCas9 protein is able to bind. And because it's binding at a key part of the promoter, this blocks expression of the output gene. And so when this was tested, it worked as hoped. So there's a range of different synthetic promoters that Adil has created for this. And you can turn off vanilla using them. And so again, it replicates the NOR gate logic. But one of the beautiful things about the NOR gate is that it is a universal gate. And so you can layer them on top of each other and achieve any of the basic Boolean logic gate combinations. And so, for example, the computer that took people to the moon had maybe 5,000 NOR gates building up that computer. And so, and that achieved all the basic computation processes that you would need to do such a complicated achievement. And so what Adil did was he took different NOR gates that he built, layer them on top of each other. And so by combining two in this format, you're able to achieve an OR gate and get OR gate logic. Whereas combining them in this combination, you're able to achieve an A imply B logic. And so I just think that this is a really nice, elegant system that is very modular. So each guide RNA is very small, so the inputs are very small compared to my very bulky recombinase genes. So it's a really nice system that I'm looking forward to more developments from that and other gene circuit works, and hopefully some applications coming in the near future. So I'm just going to wrap up now. So yeah, I just this project took a lot of work identifying the plant compatible parts to be able to build up gene circuits for both the CRISPR circuits as well as the recombinase-based circuits. Then we've been able to construct AND and OR gates, and we've also been able to construct a wide range of repressive gates and gates like the A NIMPLY B gate that mix activation and repression. And we were able to use split recombinases to build synchronous circuits where both input genes need to be turned on at the same time in order to allow for a recombination event. So that just leaves me to thank the many people involved with this. Particular thanks to Ryan Lister for building like a really fantastic environment to do research in and supporting this project and really believing in its potential, and also Florence and Patrick for being two absolutely amazing students working in the lab with me. They've been a real pleasure to have, and just leaves me to thank you.
Thank you, James. That was really great and extremely impressive for 1 in the morning. Does anyone in this audience have questions? Okay, there's one in the back. I think, do you want to try speaking? Can you hear me? Not really. Okay, I'll pass it on the microphone. Exercise.
I have a question, Margaret. Margaret, could I ask a question?
Yeah.
Hi, this is Jim, and this is great job, very elegant, and so I do have a quick question about two questions. One is, this technology is based on the Philippes. So how actually you think the completeness of that Philippes works actually? In other words, when you express this enzyme, it's all sites in the plant cell will be recombinant and loop out those like terminators and others promoters. So you think it's complete, just like you said, 0 or 1 or 1 to
yes
0?
It should be. So it acts as a tetramer to be able to cause a recombination event between the two. And in our protoplast system, we generally have what we think is hundreds of plasmids within a cell. So I don't think in the protoplast system it is going to be complete because it's got to target many, many copies of the plasmid. In a stable plant, in an ideal, homozygote single insertion line, you'll have 2 copies per cell. So we think it's likely to be much more effective. But depending on what promoter you're using as the input to drive FLP or whichever recombinase you're using, that will affect its how able it is. So with the line from the heat shock we showed, it looked like all or most of the nuclei were terminating, but we've looked at some constructs in stable lines where it does look like it's more patchy. And so I think it depends on the line. I think it depends on the promoter. I think it can, as a recombinase, it can, recombine as long as the induction is appropriate. So we did a very potent heat shock, but other input promoters may be less effective at doing that.
Okay, that's great. So another question is, related to that FRT actually sequences. So I know ATRT, what's called FRT sequence. So FRT sequence, plant genome is such huge actually genomes. By randomly, you think there are any such kind of sequences in the plant genome system that could need large scale of sort of deletion and so on and so forth?
So yeah, that's a really great question. And I think for Arabidopsis, and I have to say, I've not checked in detail for FLP and FRT sites specifically. I think it's unlikely Arabidopsis is on the smaller side of the genome. So if we're dealing with something much larger genome size, maybe wheat, for example, then it is much more likely we will have some similar sequences. However, I think the two recombination sites do have to be fairly close together within the genome for recombination to act. So I know, for example, for LOXP, I think when I was working in Moss, we figured out that I think it either almost never happened or never happened, where you had recombination between chromosomes. And so, at least being on the same chromosome is a prerequisite. And so if we insert two close to each other, they will likely recombine. But if there's a random sequence in the genome that's similar, it's unlikely that that's going to recombine unless they're very close together. And that may change with genome size, but as the genome size gets bigger, it's also, I guess, less likely you'd have two close together. So we're hoping that it'll be of a minimal issue in most plant genomes.
Okay, great.
Thank you. I'm going to pass the mic over to Randy here in the audience.
Hi, designing the gates into the plant was brilliant. And I have a question. So when designing integrated circuits, one of the problems is stray capacitance. And essentially what you have when one of the gates takes 48 hours instead of 24 hours is stray capacitance. And I'm wondering what you've thought about that and how to look for it and how to change that.
Yeah, I think you raise a really good point. And I think people are potentially going to have issues with this all over people building gene circuits. So, for example, the A NIMPLY B gate I showed you, it took 64 hours for that to stabilize and show the logic that we expected. But if you check sooner, then generally we were finding that the inhibition was slower than the activation. So again, I think based on our designs, which this is the first generation, we are seeing some of those issues, and some circuits will need more time in order to stabilize and get to the issue. Whether there's going to be some cases where we can't get the logic at all that we expect, yeah, that could well be true. And with the CRISPR circuits, we've definitely been seeing some headaches with some of the logics. So I think that could be a factor.
Do you have any hypotheses of where the capacitance is coming from? Why the time delay?
It's a good question. So in our protoplast system, I think dynamics for recombination, based on distance between them, activity speed of the individual enzyme, so whether B3 is faster than FLP, I think those are potential factors. I think what's going to be more concerning is moving into stable plants where we're adding in an extra variable, which is the promoter strength. So, if you're using a cell type specific promoter for one of the recombinases and a chemically inducible promoter for the other, then the expression level and the dynamics of that can also factor into this. So I think there's multiple things that can be causing these issues. Does that answer your question?
Yeah, you gave me two good ideas. Not that I'm going to do it, but you have two good ideas. Thank you.
Yeah, thank you very much, James. I have like one short question. So, sometimes the gene network in plants is pretty complex, and sometimes the gene expression is not like follow that 0 or 1, or yes or no, or more like the binary logic. So have you ever seen those genes that like they have like a multiple or a lot of variation or some robustness, even though you just change the conditions or something like that, it's still there. So it's still not closed, or they only just like reduce part of it or something like that. So that make those circuit, make this system like more like complicated.
Sorry, can you repeat the question?
Yeah, basically my question is, sometimes the gene is not like that easily can divide into 0, 1, 0 or 1, or yes or no, or open and close, or those like binary logic. So if like, have ever met any situation about like those genes are not, those follow that logic. So how to deal, how are you going to deal with that?
Yeah, and I think, again, this comes back to the idea of we've got to test a lot of things. So if you're trying to build, for example, so if we're talking about using an input promoter that is not, 0 or 1 to drive the input into the circuit, then it could cause some problems. Our hope is that the way these recombination circuits will work is that you might have an input promoter where one is very lowly expressed, another could be very highly expressed. But then as long as they're causing the recombination reaction, which is a one-off event, then that should lock the output gene into a particular expression level, because we're using a constitutive promoter, often 35S or actin-2. And so you're driving the output. So we're trying to create it so that the variation, of the output gene is dependent on things like insertion site within the genome. There's no way of getting around that at the moment. But we can try and reduce some of the noise from the varying expression levels from the input promoters. And I think for the transcription factor circuits, like from what Adil and Jen have developed, I'm more concerned with the variation in input promoter strengths and the variation there affecting that. So very stochastic gene expression in those cases, I think that could cause some issues with those circuits because they don't have that analog to digital conversion that the recombinase circuits allow. Does that answer your question?
Yeah, thanks.
Yes, it does. I'm over here, hopefully within camera zone. So I have a lot of ideas from your talk, but I guess just one broader question. Is there any real world proof of concept, like translational outcome that you like dream of testing now that you've set up this logic framework?
It's a great question because one of the problems we have when we're talking to people about trying to get this translated or talking about potential funding is a lot of people, a lot of companies, they're very interested in, well, what is the one application? And for us, it's like, well, we built a toolkit that can be used for almost any application. But yes, I do have some particular ones that I'm very interested in looking into. And I heard about one recently involving controlling of gene expression after harvest. So some different foods go off, different crops go off at different rates after harvest. So then, we're talking things like cabbage. And obviously putting them in the cold or fridge helps prolong them. Now, there are some ideas for how to, change gene expression that could, post-harvest, that could maybe, keep them healthier, shelf stable for longer. But do we really have a harvest-specific promoter? Most of the promoters that are activated during harvest and after harvest are going to be different stress response pathways. And I was speaking to some people last week in the UK when I was traveling there about this very problem. And, I'm very excited by the prospect of using, our recombination-based gene circuits, where you have two different promoters that are activated during harvest, both from different pathways. One could be a drought pathway, maybe one could be a very different sort of stress pathway. They would normally never be expressed at the same time. So if you just used one of them as a harvest marker, it would not be specific for harvest. But if you can combine both of them, you could make it a harvest-specific marker, and then you could manipulate gene expression post-harvest and not during those other stresses. So I think that's a potential application. But yeah, there are many more, I think, including sort of pest resistance type things.
Yeah, absolutely. Yeah,
James.
so there's one more question from
Yeah,
Susan.
James, I have one more question for you. So,
Yeah.
yeah, this is perhaps not fair to ask. And this, all your circuit, as I said, is very elegant. But the problem is one-time deal. So once turned on, then on. Once turned off, it's off. So there's no reversibility at all. So how are you going to address this issue?
Yeah, this is something that keeps coming up over and over again, and it's a very fair criticism. These circuits are very much about, one-time switches. So I think there's a few things. So one is, if you do need reversibility over memory, then the transcription-based systems like the CRISPR system and Jen's bacterial transcription factor system, they're where you want to go. In terms of us, if you want like a one-time, like you want to turn something on and then you want to turn it off separately later, I think that circuit design, we could build a more complicated 3-input circuit that could do that, where we combine activation and repression. So again, that would be a one-time done, a one-time on, one-time off, but that's possible. We haven't built that yet, but I think that would be quite straightforward. I think in terms of building a reversible one using recombinases, there are a different family of recombinases that could be used for this. There's some issues with those, but I think there is a group working on that in America at the moment. So we may see some, publications on that in the future. So that would be another approach. And then finally, where I would like to see the field go long-term would be, can we utilize these epigenetic systems within plants, like the polycomb recessive complex, like DNA methylation, to build a semi-stable, repression system where we could stably methylate a region, either at the DNA or chromatin level, and then reverse it with a demethylase. Now, the problem is that a lot of the time, we don't get good stable silencing with the current epigenome modifying tools that we have. And that's true in animals, it's true to some extent in plants. And so we need more testing in plants, I think, is the main thing. And so I think maybe one day, if we can find the right cocktail of factors for epigenome editing, we could then move away from recombinases to that. But right now, the technology is just not there. So recombinases, I think, will be a very useful stepping stone to be able to achieve that.
Great, thank you, James.
Okay, let's thanks James again. And please come visit us in Ithaca next time you're in the States.
It would be a pleasure.
Thanks, James.
No, thank you. Thank you ever so much.
This has been a production of Cornell University, on the web at cornell.edu.